Okay, thank you. Sorry about that. Yes, as Tiago said, I'm Meredith Meyer. I'm a postdoc at the University of East Anglia with Karen Haywood. So I would just like to first say thank you to Tiago, Carlos, Kate, Andrea, and all the organizers for inviting me to talk today. And I'll start by saying this is a slightly different talk than everything we've heard, because when Tiago first invited me, he acknowledged that my primary area of research in the Southern Ocean is not at the peninsula, it's actually in the Ross Sea. But I'll be presenting a study today that I think is kind of a case study that talks about some important dynamics that apply across Southern Ocean sectors. Okay, so that being said, I'll be talking, as I mentioned, about the Ross Sea, which is quite farther south relative to what's happening at the peninsula. So some of the dynamics there are quite different, and it's important to mention. So I'll just start off with a tiny bit of background for people who are less familiar. So the Ross Sea has been shown to account for 28% of Southern Ocean primary production. This makes it one of the most productive, if not the most productive sector of the Southern Ocean. And because we know the Southern Ocean plays this disproportionately important role in global primary production and global carbon cycling, the dynamics that are happening here are important on a quite large scale, global scale. The majority of that primary production occurs during the spring-summer bloom period. This is a very large bloom that is initiated in late October. It peaks in terms of biomass in mid-December and then continues through late February. During this time, chlorophyll concentrations have been shown to increase to be greater than 15 micrograms per liter. So this is a really large, a really extensive phytoplankton bloom. And an important difference between the Ross Sea and other sectors of the Southern Ocean and other oceans in general is that the phytoplankton bloom in the Ross Sea is first dominated by the haptophyte Phaeocystis antarctica. If you're familiar with Phaeocystis, it can exist as either single cells or colonies that have this sulfurous gelatinous matrix that makes it quite unappealing to grazers like krill. So it's not frequently grazed. Around late December, when the bloom reaches maximum extent, the community shifts from being dominated by Phaeocystis to being dominated by a mixed assemblage of diatoms. Diatoms are much more carbon rich, and they're much easier to be grazed by krill. So that's an important difference. And I kind of emphasize this point between Phaeocystis and diatoms because, as we know, these two phytoplankton groups are really important for Southern Ocean dynamics, but a lot of research has been shown that really emphasize the importance of diatoms and haptophytes globally. I just want to point out this study. This is a recent study from Sasha Kramer et al., where they found a really important role of diatoms and haptophytes in the North Atlantic and the North Pacific. And what they found is that when you take the biomass sum of diatoms and haptophytes relative to all other phytoplankton, so here we're looking at the x-axis, there's a really strong positive relationship between POC export flux. I mention this because it's quite relevant to the study I'm about to show, as well as just the fact that I think this is something that should be investigated in the Southern Ocean. I'd be really interested to see how this relationship plays out. Okay, so at this point, you may be gathering that a lot of my research focuses on the biological carbon pump, as shown through this schematic. So just as a reminder, the biological carbon pump is the main biologic mechanism by which atmospheric carbon dioxide enters the surface ocean, is utilized, and eventually exported to depth, where it has the potential to be sequestered on the human timescale. Given the impacts of climate change on our planet, understanding and quantifying the biological carbon pump is really important, especially in regions such as the Southern Ocean, where we don't have a particularly great grasp on what's happening. Okay, that's a very brief background because I have a lot I want to talk about today. But the study I'll be talking about is this project called Plankton 2 Predators. This was a glider-based project in the Ross Sea that occurred from 2022 to 2023. And gliders, as we as a community know, I pulled out my glider overview slide because I don't think that's necessary for this group. But as we know, they've been really, really helpful in expanding our knowledge, particularly of regions such as the Ross Sea, which is traditionally understudied. But that being said, the Ross Sea is so south and so logistically difficult to get to that despite this, despite the importance of gliders and the impact they've had, there's still very few studies that exist, particularly when we're talking about gliders that are equipped with biogeochemical sensors. However, Karen's group has been involved in 3 of these studies. The one that I'm talking about today, as I mentioned, occurred from November of 2022 to January of 2023, where we deployed 2 sea gliders from the Ross Ice Shelf Fast Ice, and they surveyed in this kind of grid-like pattern, coinciding with the peak and beginning of the dissipation of the Ross Sea phytoplankton bloom. Okay, so what did we find? Here we have a figure from Portela et al. Esther was the original postdoc on this project. I was the secondary, or I was the one that picked up when she left. But so here we have 20 years of reanalysis data from ERA 5, where we have PAR anomalies on our top plot, sea ice concentration anomalies in our middle plot, and then wind speed anomalies on the bottom. And what we found is that in our year of interest in 2022 to 2023, we had anomalously low PAR and wind conditions and anomalously high sea ice concentrations. During this year, we know that the Ross Sea polynya, which generally controls the timing of the phytoplankton bloom, opened late. So this led to a really cold, really icy season. And this had very noticeable impacts on our biogeochemistry. So here we have some glider plots from this study where we have date on our X. Our top plot is the distance to sea ice concentration, and then we have temperature, salinity, chlorophyll A being a proxy for phytoplankton biomass, POC or particulate organic carbon, also a proxy for phytoplankton biomass, and then the ratio of carbon to chlorophyll. And so what we've noticed right away is that this year appears to have had substantial biomass accumulation. And this is most obvious by chlorophyll concentrations that were greater than 40 micrograms per liter. So this is really massive. This is really unprecedented. Interestingly, though, we had lower than average POC concentrations with values about 200 micrograms per liter. And I think a lot of this has to do with this shift in our taxonomic balance. So higher chlorophyll and lower POC tends to correspond to a presence of phaeocystis. So overall, I think what was happening this year is that we had more phaeocystis and less diatoms than normal. Additionally, so we have a lot of biomass, a lot of predominantly phaeocystis dominated biomass, and we see that that's hanging out in the surface waters. So here we have, again, date on our X, and then we have our average POC concentration at various depth thresholds through time. And what we see is that in the ramping up and peak of the bloom, we have this consistent pattern across these different depth thresholds. But towards the end of the bloom, we see this uncoupling where we maintain high POC concentrations in our upper 25 meters, but at 50 meters and below, we see a decrease. So not only is there a lot of biomass, but it appears to be hanging out. It doesn't appear to be removed in the same way that was happening earlier. So I've been talking a lot about POC and chlorophyll because these are important biogeochemical stocks that we tend to use as proxies for rate processes that are happening. But we do have the ability to explore the rates themselves. So in this analysis, I was interested in calculating NCP, or net community production, which is just our total production minus our total respiration. And we can also calculate our change in POC over change in time. So this gives us this kind of localized export measurement. And then if we take that and subtract it from our NCP, it gives us our POC export potential over a longer time period, over the annual scale. So what did we find? Overall, we found that in 2022 to 2023, when we had a lot of biomass, we also had a lot of NCP, but we have this relatively low and invariant rate of change in POC over change in time. So this was quite interesting, but to try to understand what this means in the grand scheme of things, we took all of these stocks and rates and physical properties and compared it to 2 other years of glider data. So in Karen's group, we also have campaigns that occurred from 2010 to 2011 and 2012 to 2013. And I don't have the time to get into this plot in too much detail, But what I want you to notice is that across these different years, we have the colors vary from plot to plot. So we have important differences in our physical properties, our temperature and salinity, a lot of which correlates or corresponds to changes in mixing as well as changes in ice presence. And then we have interesting differences between our biogeochemistry. So, for example, in 2012 to 2013, we had really high POC concentrations, much higher than we're seeing in 2022 to 2023. We can also take all of this as well and look at our rate measurements across our different years. And so what we found is that in all years, we had high but variable rates of NCP. And in all years, we had pretty low and pretty invariant rates of change in POC over change in time. So this led us to conclude that the Ross Sea falls into this export production regime of high primary production and low export. So this is a very, very quick overview. But what this study found was that in 2022 to 2023, this was a really anomalous year, both in terms of chlorophyll and ice. So we believe that's related. We believe that the ice conditions changed the phytoplankton community, leading to a large accumulation of biomass. Another important thing is that we found there's this uncoupling between production and export, where we frequently have high production and low export, but there's also this interesting uncoupling between our biogeochemical stocks and rates. And this gives us a measure of our not only ecosystem efficiency, but it's an important consideration when we talk about scaling up these dynamics, specifically for models and just large scale analyses. So what I mean by that, because I only touched on it briefly, is that in certain years, such as 2012 to 2013, we had a lot of POC, but little NCP. Whereas in 2022 to 2023, we had little POC, but a lot of NCP. And in 2010 to 2011, we had little NCP and little POC. So this lack of consistency between years means that it's not quite as simple as we frequently like to think it is. We can't always assume that a lot of biomass equals a lot of production and a little biomass equals little production. There's a lot of intricacies that are varying based on physical conditions. So this is just something we as a community, I think, need to think more about when we frequently use these biogeochemical stocks as proxies for rates. Okay, and then taking all of this and putting it together, I want to look forward. So in this analysis, we think the change in sea ice concentrations played a big role in setting changes in production and phytoplankton community. So that would suggest that as conditions change in the Ross Sea, there will be similar effects. So more ice equals more production, less ice equals less production. That's obviously a very large simplification of what's happening, but that's what we're seeing. The interesting thing is that we're not necessarily seeing the same degree of change in our carbon export. So in a sense, what's happening with the ice is less important for what's happening in carbon export. And this is a really thought-provoking finding because this has some interesting implications when we're trying to build a Southern Ocean carbon budget and just explore Southern Ocean biological carbon pump dynamics in general. Okay, so that, as I said, is a very brief overview of what I'm think we can consider here as a case study, but I want to spend the remaining time I have and just bring this back to the peninsula. And so specifically, I want to return to the Weddell Sea through the Piccolo project. So if you were here on Tuesday, you may have heard the lightning talk from Isabel Seguro, where she briefly overviewed some dynamics from the Piccolo project. Piccolo was a large UK-based project that involved a cruise in 2024 to the Weddell Sea. This was led by Karen Haywood and Tom Bell, as well as a number of collaborators. And then here is a cruise map from the Piccolo campaigns. So we're over here. And this project has a ton of data, but the data I want to show you today is some HPLC data. So here we have some HPLC from the Larsen Sea ice shelf region. And I want to draw your attention to the green bars here. So these bars correspond to 19-hex fucoxanthin, which if you are familiar with HPLC, you'll know that this is an accessory pigment that we frequently use to diagnose the presence of haptophytes. And haptophytes are a phytoplankton group of which Phaeocystis is a part. We know from other data that there was a large Phaeocystis bloom at the Larsen C ice shelf region during this time. So we have pretty high confidence that this 19 hex corresponds to Phaeocystis. Another interesting thing to point out is that we have both deep and shallow samples here, and we have this fairly consistent high presence throughout, regardless of where we are in the water column. So this is quite interesting. During the Larsen C sampling, this is also where we had the highest concentrations of biomass during the entire Piccolo cruise. And one thing I want to point out is that the region was really ice covered up until it was sampled by the cruise. So basically, the participants realized that the Larsen C ice shelf region had been uncovered by ice for a couple of days, quickly ran over, sampled the region, and then quickly left before the ice returned. So we have a pretty strong confidence that what was happening here was the result of this ice melt. So what I think happened is that the ice melted, it seeded the water with trace metals and alleviated light limitation, allowing this Phaeocystis bloom to occur. And there's some interesting things to think about when we think about this Phaeocystis bloom, given what I was showing in the Ross Sea. Oops, now go forward. Okay. I know I'm rapidly running out of time. I may even be over. Sorry about that. But because I'm about to end, but one of the things I wanted to end with is just kind of some thoughts. Since the SOOS workshop is a fairly open format, I think it's beneficial as a community to kind of leave everything with some lingering questions. So here are my kind of last thoughts and open-ended questions. I've been talking a lot about the impacts of physical parameters such as sea ice on biogeochemistry and changing taxonomy. And I'm definitely not the only person to be interested in this. Here is a recent paper from Alex Haywood that came out a few months ago, where he looked at changes in diatom, haptophyte, and cryptophyte abundance across the Southern Ocean and related it to a number of physical parameters, specifically sea ice melt. And he found some really interesting things. So I think this is a great jumping off point for us as a community to start to consider how these dynamics change and how it relates to carbon export, primary production and carbon export potential. As I mentioned earlier, there is some interesting implications. So here's a figure that I love from one of Steph Henson's papers in 2019. And based on our current findings in the Ross Sea, we put this region in this high primary production, low export, largely related to this role of Phaeocystis within the community. Now, if the balance between different phytoplankton groups is changing in the Southern Ocean through time, there's a lot of interesting questions that arise and implications for what this means. I think it's worthwhile to consider the different sectors of the Southern Ocean, where they fall within this table, and how this will change geographically, as well as the role of interannual variability in these dynamics, and especially how these dynamics play out on continental shelves, where we tend to have less data and where things tend to be a bit more dynamic. So I will leave that there with a bunch of open-ended questions, and I'm really happy to talk about this with everyone. But yes, thank you for your time today.